Hello everyone, my name is Simran Bawa. I am a PhD student at Kansas State University, and today I will be talking about new identified role of Drosophila TRIM32 in promoting cell growth. To begin with, what is TRIM32 protein? TRIM32 protein is a member of tripeptide motif family identified by the presence of ring domain, B-box domain, and coil-coil region at its N-terminus, followed by 6 NHL repeats at its C-terminus. All these domains, they have independent functions. For example, ring domain imparts E3 ligase activity to the protein. Because of the E3 ligase activity, TRIM32 can add multiple ubiquitin molecules to the target protein and can specify their function. Likewise, NHL repeats are known to mediate protein-protein interaction. NHL stands for NCL1, HT2A, and Lin41. Because of the sequence similarity these repeats share with the proteins. Another interesting thing about TRIM32 is that it is ubiquitously expressed. Because of the ubiquitous expression of TRIM32 protein, it has function in muscle physiology, regeneration, and cancer. For example, under normal cellular condition, TRIM32 can polyubiquitinate c-MYC and XIAP, which is inhibitor of apoptosis, and can induce apoptosis or cell death, and also can control the differentiated state of the cell. In a way, it acts as a tumor suppressor. However, under abnormal cellular condition, such as in case of cancer where TRIM32 is overexpressed, TRIM32 can polyubiquitinate tumor suppressor proteins like ABI2, P53, and PASI, and can cause increase in cell growth and reduction in the cell death, and can have oncogenic role. TRIM32 is also known to regulate protein turnover of some of the muscle proteins like actin, desmin, and dysbindin, and plays an important role in maintaining normal muscle homeostasis. It is also implicated in one of the muscle disorders known as limb-girdle muscular dystrophy type 2H. All these functions have been identified and studied before, but today I'll be focusing on telling you all the role of TRIM32 in regulating tissue growth. For my research, I use Drosophila melanogaster as a model organism because Drosophila also possesses homologue of TRIM32, which is highly expressed in muscle. So back in 2011, our lab published a paper where we showed that loss of TRIM32, also known as thin in Drosophila, results in muscle degeneration. As you can see, muscles appear thinner and they have lost their structural integrity and have unbundling phenotype. However, when we did the rescue experiment where we put back TRIM32 full-length cDNA using a muscle-specific promoter, we were able to rescue the defects. Later on, in 2013, another group tried the same experiment where they did the rescue, however, without the NHL repeats, and what they found was that rescue experiment performed with just the N-terminal part of the protein did not rescue the defects. The muscle still appeared degenerative in nature and they had lost their structural integrity. So this experiment actually provided evidence that these repeats are really important and they play an important role in preventing muscle degeneration and are required for normal TRIM32 function in the muscle. Since the overall goal of our project was to identify new substrates that bind to TRIM32, and we know that NHL repeats mediate protein-protein interactions, so we just focused our study on the C-terminal part of the protein. So I expressed the C-terminal part of the protein and fused it to GST, and I immobilized the protein onto the beads, and then I incubated the protein with L3 larval lysate, and then I eluted the protein which is bound to the fusion protein of the complex, and then the complex or the bead complex or the eluted protein was sent off to Oklahoma State University for mass spec analysis. So this is the table that summarizes our result that we got back after our mass spec analysis. So the very first thing that we want to make sure that our experiment was a success is to find out that whether we were able to recover our bait protein or not. So yes, bait protein, which is TRIM32 NHL, was present only in the experiment but not in the control, and to our surprise we also found some of the glycolytic enzymes were enriched in the experiment as compared to the control, and few of them were not present in the control at all. Another think that another protein that we identified out of the mass spec data was tropomyosin 2 and troponin T, which are the known targets of the human TRIM32 protein. So this was really comforting because this provides strong evidence that two proteins, although from different species, they are functionally conserved. The very first thing you want to do after you get your mass spec data is to validate it. On the left is the schematic of glycolysis, where all the enzymes highlighted in blue were enriched in our experiment, and we decided to validate the binding of two of the glycolytic enzymes, aldolase and Phosphoglycerate, with TRIM32 NHL protein. I expressed aldolase TRIM32 NHL protein and a control protein to perform the binding assays. I immobilized aldolase in control onto the nickel NTA beads, since both of the protein they have N-terminal His tag, and I then incubated TRIM32 protein with aldolase and control protein. As you can see that TRIM32 only binds to the aldolase and not to the control. And similarly, we observed the same result with Phosphoglycerate, where TRIM32 only was bound to the Phosphoglycerate and not to the control. So our binding assays actually validated our mass spec data that TRIM32 directly interacts with two of the glycolytic enzymes, aldolase and Phosphoglycerate. Since TRIM32 directly binds to two of the glycolytic enzymes, we decided to do metabolite profiling on our TRIM32 mutants and used wild type as a control. We sent a frozen larvae in liquid nitrogen to the University of Utah, where they extracted the metabolites using GCMS and used mass hunter quant software to identify the metabolites and did statistical analysis on it. On the right is the volcano plot, where y-axis is the log p value and x-axis is the log fold change. The vertical line represents the fold change and the horizontal line depicts the p-value. And to our surprise, we found out that two of the terminal products of glycolysis, which is pyruvic acid and lactic acid, were significantly reduced in our TRIM32 mutants, which can also be seen in our whisker plot, where lactic acid and pyruvic acid, they're relatively less abundant in the mutants as compared to the control. We also analyzed overall ATP levels and found out that in TRIM32 mutants, the levels were significantly reduced as compared to the wild type. Therefore, the metabolomic analysis provides evidence that TRIM32 is required for maintaining glycolytic flux in the animals. As I have previously mentioned, that loss of TRIM32 results in thinner degenerative muscles, and we always thought that this loss of muscle mass is a consequence of muscle degeneration. However, the data we got from our metabolomic analysis and our mass spec pointed us in a different direction. As you can see in our wild type, as we go from L2 to L3 larval stage, there is massive increase in the size of the tissue, which is also depicted here in a scatter plot. You can see a massive difference. However, in our TRIM32 mutants, as we go from L2 to L3 larval stage, there is no increase in the size of the tissue. Therefore, we specifically decided to look for glycolysis in our TRIM32 mutant muscles. And to do that, we used ECAR assay. So ECAR is basically extracellular acidification rate, which measures the excretion of lactic acid upon glycolysis. And we find that glycolysis was significantly reduced in our TRIM32 mutants as compared to wild type. Hence, our ECAR data along with our metabolomics data confirms that in our TRIM32 mutants, glycolytic flux is significantly reduced. Just like larval muscles, Drosophila larval brain is highly glycolytic in nature. As you can see on the left, we have used LDH-EFP reporter to look at the endogenous glycolytic activity. You can see high glycolytic flux in the larval muscles, and similarly you can see it in the larval brain. We have so far established that TRIM32 mutants exhibit reduced glycolytic flux, and specifically TRIM32 mutant muscles, they have reduced glycolysis. Therefore, we wondered whether TRIM32 can also affect other tissues which are highly glycolytic in nature. So we decided to analyze mutant brains. Drosophila larval brain comprises of 2 brain lobes and 1 ventral nerve cord. By just looking at the image, you can see there is significant reduction in the size of the TRIM32 mutant brain as compared to wild type, which is also depicted here in a scatter plot. When we measure the brain area, we find that there is significant reduction in the area of the mutant brains. So just like muscles, we decided to analyze glycolysis in the larval brains, and we made use of PER assays. PER stands for proton efflux rate. which measures the protons that are being produced both by mitochondrial cellular respiration and through glycolysis. So the first few cycles over here are the basal measure of protons that are being produced by both cellular respiration and glycolysis. Since we were interested in analyzing protons that are being produced through glycolysis, we made injection after 7th cycle using rotenone. It's a drug that inhibits mitochondrial function, and we see a sharp increase in the production of protons in the wild type as compared to that of the mutants. So now we wanted to be sure that the protons that are being released are through glycolysis. We made another injection after 12th cycle using 2DG, which is an inhibitor of glycolysis. So if the protons that we see are being produced through glycolysis, we should see a sharp decrease in the protons production, and that's what we observe over here. Therefore, our data provides evidence that indeed glycolytic flux is reduced in our TRIM32 mutant brains as well, just like in the muscle, and also they exhibit smaller brain size. There are a couple of reasons that can contribute to reduction in the size of the tissue. 1, there are not enough cells that are proliferating. 2, there is increased cell death. 3, simply cells are not able to accumulate enough biomass in order to grow in size. So we first decided to look at the cell proliferation, and we made use of EDU incorporation assay. So EDU is a thymidine analog, and it gets incorporated in cells that are actively synthesizing DNA. So all these green little dots that you see over here are the cells that are actively synthesizing DNA. So we measured these EDU positive cells, and we find out that TRIM32 mutant brains are not defective in cell proliferation. We also looked at cell death, and we did not find any changes in the wild type and TRIM32 mutant. Therefore, the two assays that we did suggest that an increase in cell size is the primary mechanism by which TRIM32 mediates growth of the tissues. The elevated glycolytic rate that operates in larval muscle and brain tissue is analogous to the Warburg effect, where the proliferating cancerous stem cells utilize glucose metabolism for the generation of amino acids. Therefore, we hypothesized that maybe TRIM32 could be a general regulator of highly glycolytic tumor cells. We made use of Drosophila PVR wing disc tumor model. PVR is shown to activate canonical RAS-MAP kinase pathway, and it has already been shown that expressing activated form of PVR in Drosophila wing disc using DPP, which is a wing disc driver, results in a massive growth of the wing disc and induction of LDH, which is a hallmark of aerobic glycolysis and the Warburg effect. We decided to analyze the effect on wing disc tumor in our TRIM32 mutant background. First, in general, wing disc is not glycolytic in nature, so in our control, we do not see any LDH activity. When we express the activated form of PVR in the wing disc using DPP as a driver, we see massive growth of the wing disc, and also we see robust LDH activity. However, when we express the activated form of PVR in a TRIM32 mutant background, we see a significant reduction in the size of the wing disc, which is also depicted here in our bar graph, where you can see in general wild type and TRIM32 mutant wing discs, they are comparatively of the similar area. However, when we express the activated form of PVR in the wing disc, we see a massive increase in the size. And when we express the activated form of PVR in TRIM32 mutant background, we see a significant reduction in the size of the wing disc, and about 50% of the wing discs in a TRIM32 mutant background showed LDH expression. So the data confirms that overexpression of PVR in TRIM32 mutant background reduces the size of the tumor. Our current working model is, in the presence of TRIM32, glycolytic enzymes such as PGLM and aldolase bind to the NHL repeats and in turn increases the glycolytic flux, which provides intermediates which are required for the production of nucleic acid, lipids, and proteins, which in turn is important for the biomass accumulation and helps in the growth of the tissue. However, in the absence of TRIM32, there is no binding of the glycolytic enzymes. There is a reduction in the glycolytic flux, which in turn reduces the cellular biomass and which has effect on the cell growth. With that, I would like to thank my advisor, Dr. Erica Geisbreck, all my lab members, committee members, collaborators, and the funding for making this project possible.